Yeah, please, if you could introduce. Yeah, Pushpendra. Yeah.
Good morning, everyone. It's my pleasure to introduce Dr. Muhammad Ilyas, who is assistant professor at, uh, as far as I know from his profile, he is working at the Center for in fact, he has met some of the very well known and reputed centers globally, such as University College of London, uh, in Harvard also, and several reputed institutes in Korea, also in Hazara University and CEMB Pakistan. So his work has a lot of diversity actually, and he has been conferred a lot of reputed awards and scholarships throughout his career, including Best Young Scientist Award, Best Research Paper Award, and what struck me most was that just like the diversity of bioinformatics, he has worked in so many different diseases and topics such as HIV, hepatitis C, hepatitis B virus, dengue virus, cellular protein, whole genome sequencing of Pakistan, individual from Pakistan, as well as for the evaluation of synthetic hexaploidy of beets. In addition, he has also worked for the E. coli fatty acid and biodiesel aspect of biology, and also for the in silico modeling of Indian Congo hemorrhagic fever virus. So with that, I would say that he has actually very good example of what we do in so many different fields, but just as a single individual, he has contributed in so many fields that is very much inspiring. I would simply and summarily say. So with that, and I hope there is some disturbance due to the background noise during this introduction. So I would hand over to Dr. Muhammad Ilyas to present his talk on genes and genomics in Pakistan. Thank you very much, and it has been a privilege to introduce you. Thank you very much.
Thank you, Pushpendra. Thank you so much. Prash, can you see my slides?
Yes, please. Yeah, can you make it full screen, Pushpendra, please? Yeah. Sorry, Ilyas. Yeah. Can you please make it full screen? Yeah.
Is it okay?
You'll get a tab over there, Ilyas. tab click करने से आपको क्या है एक और लिंक आएगा उसमें अगर आप तब फुल स्क्रीन आ जाएगा आप एक बार देखेंगे प्लीज
I'm trying to do it.
Yeah, otherwise if you can quickly log in again and just refresh your online browser. कभी कभी Google Chrome ये problem देता है if you can kindly, yeah, please.
Meanwhile, I would just say that it's really he has also helped in developing this field in Pakistan. That are you able to click on the full screen button, Dr. Ilyas?
Sorry, I can't see the full screen button.
Yeah, it's okay, Ilyas. Agar aap, can you move to the next slide so that we can see if it is moving or not, if it is transiting or not.
When?
Can you please move to the next slide? Yeah, it's moving. It's moving. Yeah, you can start. Yeah, I think you can start. Yeah.
Moving now? Ash?
Yes, very much. Yeah, yeah.
Okay. Thank you so much. For inviting me. So I'll be talking about the current status of genes and genomics in Pakistan. So what we are doing here in my institution and the other institutions as well. I'll just give you a brief introduction. My name is Muhammad Ilyas. I'm assistant professor at the Islamia College, Peshawar. It's in the Khyber Pakhtunkhwa province. Islamia College was established in 1913. It's a public sector university, and it was granted university status in 2008. We have 4 different faculties and 23 academic departments and research centers. This is a visual of Islamia College Peshawar. You can see that it's actually it has some historical background as well, and it's rooted with Aligarh University. I'm working in the Center for Omics Sciences, which was established in 2017, and we offer MPhil and PhD admission once a year in the field of genetics, molecular biology, microbiology, and bioinformatics. So we have different research groups. I'm leading the genomics and biomedical informatics lab. We have forensics and molecular anthropology lab research. A group is working on rare genetic disorders. One of our colleagues is working on cancer biology and stem cells, and we have plant pathology and food security group as well. This is a 3D model of our research institute, which is going to complete in the end of this year, and we will be shifting our activities to this building. So it has different labs and facilities. The Pakistani population is about 241 million, and we have a literacy rate of 62.8%. This is different distribution of the 4 provinces. We have 75.5% education in literacy rate in urban areas and 58.8% in rural areas. 73% of our males are educated, and 51% of our females are educated. We have about 800 different educational institutions, and you probably you may know about these people. All these are have got their education from Pakistan. Khurana, who is from India, and he got the Nobel Prize in 1968, and he got his basic education from University of Punjab, Lahore. Abdul Salam, who was a Pakistani Nobel laureate, and he also got his master from University of Punjab. Malala Yousafzai, who recently got Nobel Prize in Peace. So she is from Pakistan, and she is living in my neighbor in Swat Valley. I'm from Swat Valley, so we are living in neighbors. So Malala Yousafzai is basically my neighborhood. So all these people have some interaction, some connection with me in some aspects. Like the 2 guys got their PhD, their education from Punjab University, and I did my education, my MPhil and PhD from Punjab University. And Malala is my neighbor. So one more thing, one more interesting thing which I want to mention here is that I work under the supervision of Jong Buck, and Jong Buck work under the supervision of George Church. George Church was actually a student of Gilbert, and Gilbert was a student of Abdul Salam. So we have some academic genealogy as well. These were just some interesting facts that I wanted to share with you. So we have about more than 250 universities who are offering different degree programs in different fields. And we have 58 public sector universities in Punjab, 34 public sector universities in Khyber Pakhtunkhwa, 9 in Balochistan, 15 in Sindh. Some of the universities are in the top ranked from Pakistan. These universities are in the top ranked on the basis of their research activities and their academic activities. Coming to the field of bioinformatics, bioinformatics was started in Pakistan in 2003 in these 4 institutions: Islamic International University, GC University Faisalabad, Muhammad Ali Jinnah University Islamabad, and ComSat Islamabad. They were the pioneers to start the field of bioinformatics in Pakistan. And in 2011, I published a paper in PLOS Computational Biology, and in that paper, you can see a graph that will show you that that time in 2011 there were about 10 or 11 institutions who were offering bioinformatics degrees, but now we have about 38 universities who are offering BS degrees in bioinformatics, and about 12 or 14 institutions who are offering MPhil or MS degrees, and 5 universities are offering bioinformatics in PhD level. While I was student in MPhil PhD, we started a society who was trying to develop bioinformatics in Pakistan. So we organized different events, different workshops. So we are going to arrange some online sessions, and that was I guess 2008, 2009. So then I met one of my friend in South Korea, Rohit Reja. He told me about the ISCB and the RSGs affiliated with the International Society of Computational Biology. So then we registered ourselves with the ISCB as RSG Pakistan, and there were about 20 or I think 20 different RSGs, regional students groups from around the world. And in those days, in 2010 or 11, I met with Prash, and Prash then helped me promote and develop the bioinformatics and the awareness about bioinformatics in Pakistan. So whenever I had some issues, I discussed it with Prash and Rohit and other fellows from India. So they helped me a lot. So after a few years, then we established a society, an independent society on a university, on a large level, on a national level. So that was Pakistan Society for Computational Biology, and that society was running till 2020, I guess. So we organized many workshops, many internships, many conferences. So bioinformatics in Pakistan has different potential areas. Students who graduated from these fields or from these universities getting their degrees in bioinformatics, they are currently working in software development area. They're developing databases on different issues and different data sets. They're analyzing the data, and they're working in different research groups. Like we have different research areas in plants, in microbes, in animal sciences, and human sciences. So they're really working very well in these fields. And most of the people who are trying to develop their skills they are moving forward, and they want to get the advanced knowledge, the advanced skills in the field of bioinformatics. They move to other countries like US and UK to get their scholarships and to get the additional education. So many of these students, many of our students have started their own businesses as well, and they developed different startups. They developed some apps, mobile apps, some companies these companies are working in health science, in healthcare field, in crop field, in microbiology fields. Two of my students have developed apps. One was a Najumix, and the other was Mediomics. So they developed that tool to predict the personalized medicine using the genetic makeup of an individual. So I started bioinformatics. I joined the bioinformatics field in 2008 in Center for Excellence in Molecular Biology. Then I moved to South Korea for MPhil and PhD. Where I joined KOBIC, Korean Bioinformatics Institute, and I worked with Jong Bak. He was my co-supervisor actually. And then in 2015, I joined Harvard Medical School and I joined Precision Medicine Lab, where I worked with Peter Tony Letos. He was my supervisor. So coming to our research activities, actually it started with the Human Genome Project. So when the Human Genome Project completed in 2003, many other institutions and groups have started sequencing the other genomes as well. In 2008, a new era of personalized genomics have been started, and different ethnic genomes were sequenced in all over the world: Chinese, Africans, Indians, Koreans, Sri Lankans. So in 2013, the first Pakistani genome was sequenced by HEJ Karachi. They sequenced the first Pakistani and the first Sindhi genome. Then in 2015, I myself sequenced the first Pathan genome, or you can say Pashtun genome, in collaboration with Jong Bak. I did my work with him in his lab. So we sequenced and we reported this genome in 2015. At that time, we had only one available genomes from Pakistan, and that was the Sindhi genome. So I was the second one to sequence the first Pathan and the second Pakistani genome. So these were the results. When we compared our result, we analyzed the result. The first question was what we have to do with the genome. When we sequence the genome, what were the objectives of this project? So we were trying to check the ancestry or the genealogy of the genome. We were trying to use this genome for disease risk, to identify the disease risk in this individual, and also we were trying to find a workflow that would help us in designing the personalized medicine as well. So when we compared the Asian genome, we found we got a green chromopanther, and when we run or we analyzed the European or Caucasian genome, we got the red chromopanther. But when we analyzed our Pakistani genome, we found both colors in an individual. That was because the Pakistani and Indian and South Asian genomes are admixed populations. So we had different genomes, whole genomes available online, like at that time 1000 genome project first phase was released. So that the panel had Eurasian African, Caucasian, Indians, Koreans, and Chinese populations. So we compared our sample with those genomes, and we found that our sample had similarity with the Indian genomes. So we found we came to the conclusion that this individual is a South Asian genome. In case we had no clues of the individual, we can say at this stage that this individual is from South Asian region. Then we compared this individual with the already available data sets which was released by Harvard University. David Rick released that data. The project name was HGDP, Human Genome Diversity Project. So it was not a whole genome project. They didn't have whole genomes, but they released microarray data sets which had about 6 million, I guess 0.6 million SNPs printed on those microarray chip. So we compared our sample with those samples, and in that data set we had different ethnic groups from Pakistan, like Bodhi, Punjabis, Balochis, Sindhis, and some other populations. So when we compared our sample with that data set, we found that this individual is exactly a match of the Pathan genomes, which was released by HGDP. Then we did some MDS analysis as well, and we found the same result. We got the same results. On the in the blue color, you see a workflow. This was the pipeline we developed for analyzing these kind of studies. So you can use this pipeline for personalized medicine or personalized genomic studies. In another project which I was running, it was about the Pakistani ancient and modern DNA samples. So we collected some samples from different regions of Pakistan. We collaborated with museums and archaeology department, and we collected those samples. We brought it to the lab. We extracted the DNA, and then we did the mitochondria and the Y chromosome analysis. So we unfortunately we were unable to study the Y chromosome and the autosomal chromosome just because of their quality. So we did only mitochondria DNA analysis, and we found that the M haplotype is the most common haplotype in mitochondria, in Pakistani mitochondria. We also did, and in another project, we used the modern Pashtun population or the Pathan population. The Pathan population is distributed in different other ethnic groups as well. So one of the biggest ethnic group in Pathans is Yusufzai. So Yusufzai migrated from Afghanistan to Khyber Pakhtunkhwa or to Pakistan. So we sequenced their Y chromosome, we genotyped their Y chromosome, and we found that the R1A haplotype is the most common haplotype in Yusufzai or in Pashtun population. So by using this haplotype, you can identify the ancestry of a population. The other study, the Y chromosome, you can see the Y haplogroups here. So this is actually this was published in Nature by Harvard University, David Reich group. So they used samples, ancient DNA samples from Swat regions, from Khyber Pakhtunkhwa Swat region, and in those samples they identified that L is the most ancient haplotype in the Pakistani samples, or you can say in the population living in the northwestern areas. You may know that Pakistan is at high risk or has a high frequency of cousin marriages. So that's why people around the world, all the researchers working on the genetic disorders or the ancestry or the genetic characterization of different diseases, they're interested in Pakistani population and specifically Pashtun population, just because they have the high frequency of the high percentage of the cousin marriages. So that's why we used cousin marriages, and we are trying to identify the genetic characterization, or we are trying to find out the genetic variants that are associated with different diseases. So my other colleagues are working on different diseases, but I have focused on only neurological disorders. So I have a grant I received from Higher Education Commission from Pakistan. So in this project, we almost collected 200 plus families that has neurological disorders. So far, we published about 3 novel genes and more than 100 novel variants. We sequenced almost 200 patients, and we did whole exome sequencing of those sequences. The circle graph, the Circos plot, in the bottom, you can see there are different labels, and the label mentioned all the sites, all the positions that we have reported, all the variants that we have reported in the Pashtun population, and the black spots presents all the SNPs or all the variants that are present in a Pashtun, in a Pathan individual. So since 2021 till now, we have published a number of publications in different reputed journals, and if you see one of the journal, Congenital Anomalies, they published our papers, and then they published it on the cover page as well. So it was basically a big achievement for us, or a big honor for us. So they publish it as a cover page. These are different genomes that have been published from Pakistan. The ethnic genomes: Punjabis, Pathan, Sindhi, Punjabi, Kailash, and some ancient samples as well. Right now, if you search the SRA NCBI, you will find out that there are more than 30,000 genomes, exomes, and whole genomes published from Pakistan that are available on SRA. So we have a lot of data right now to do some additional work using bioinformatics skills. At that time, at that time when we were doing the whole genome sequencing, or the Pakistan Genome Project, we collaborated with different institutions. Like HEJ collaborated with China, I myself collaborated with South Korea, and there were other institutions that collaborated with other organizations like John Hopkins and Wellcome Trust. Right now, we have our own machines in different regions in Pakistan. We have about 15 MI6, and some of them are Hi6, and we have Sanger sequencing. So we are now sequencing our own genomes by ourselves. So the main problem that we were facing during our research was data collection. So when we were collecting the samples, either it was for ancestry study or it was for the disease risk, we faced a lot of problems to identify and to collect the sample. So we developed a mobile app that could help us in collecting the samples. Like if someone install it in their mobile, they can contact us. They can submit the data of an individual. If they met someone on the street, they can take their pictures and submit it on this app, and we can identify the location. We can contact the individual. We can contact the patient family, and we can ask many other questions regarding their disease. And if we are interested in collecting their samples, we send our team, and then they collect the blood samples and other phenotypic information. They get the consent form, everything, and we bring the sample to the lab for further studies. In addition to that, we have already developed a database, Pakistan Genetic Variation Database. It's a very important aspect that all the variants that we have published by myself or many other individuals around the world are in Pakistan. We collected all those variants, and we developed a database. So everyone interested in human genetics are in variomics of Pakistani population, so they can use this database and get the basic information, or they can request us to get the whole database data set if they're trying to move on for diagnostic studies, or they're trying to use these data for precision medicine. So we can share this data with them. In addition to this, whenever we met the patients or their families, they always ask about the productivity or the result of the project. Most of the people doesn't even know what is genetics, and how can they help. How can this genetics help them in their cure or in their therapies, or developing some medicine. So we published some books in Pashto, in Urdu, in English. It's already available online. but we publish these books for our people in Pakistan. So we share these books with the people, and to make them understand what is genetics and what we have identified so far, and where are we going. So these kind of books are really helpful for these people. We also organize different events in Pakistan in different regions. In fact, we did try to make our world record by collecting more than 1,000 students, and they isolated the DNA sample at one spot, at one region. We regularly organize DNA Day every year, DNA Day and another Rare Disease Day. So we organize these events every year for the awareness and for the feedback of the people. I'm thankful to all the people, the organization, the groups who are helping me in running these kind of projects. This is my whole team. Sheriar, Ayaz, and Taurahman, they are my PhD students, and the other are MPhil and MS students. Some of them have graduated from my lab, and right now they're working in different colleges, universities, and different health departments and serving their country. Thank you so much.
Yeah, thank you so much, Ilyas, for a very scintillating talk. Pushpendra is traveling, so I'm not sure if his connection is good. So if I can take on his behalf in thanking you in person, for this wonderful talk. Friends, any questions for Ilyas, please? Yeah. No, in fact, Ilyas, stole the show. In fact, he also, put forth, wonderful instruments. That was, the real, key for, Pakistan development in bioinformatics. And, we have, very few people, who return back to their, country and, ensure that, their particular science that they have learned, is taken up, to the higher level. And Ilyas is one among very few who has been taking pains for for development of bioinformatics in Pakistan. Thank you so much, Ilyas, for the wonderful talk. Yeah, yeah. I think we will please please stay. We will have we will have something in panel discussion. On crosstalk and also collaborations. Between South Asian network countries. Yeah. Any questions for Ilyas, please? Yeah, friends. Yeah. Yeah, yeah, I think there is someone who has raised the hand. I'm sorry. Who is that? 1 minute. Yeah, yeah. Darshini, please. Why don't you please talk, please? Yeah. Why don't you
Hello.
raise your question?
Hello.
Yeah, please.
Yeah. So it was a very nice session. And the only question I want to ask is you talked about an app that was developed like Genoloca. I want to know that what was the response you got and how did you collect those information? I mean, for the genetics. What was the response you got from the locals or the sources from which you collected the data?
Thank you. Actually, we launched that app for a few days, and we got a very good response from the people, especially from the doctors. That they contacted us and they were asking for more help regarding the genetics involving in medical studies. Later, we stopped the app just because some kind of errors. So we will be releasing it again in next month. I hope so.
Oh,
So the response was very good, and we get a list of patients and list of information from the people. Most of the people are interested in genetic studies.
Okay, thank you.
Yeah. So thank you so much. Keeping in view of brevity of time, so we are just 5 minutes delayed or 6 minutes delayed for that matter. We have Asif's talk, but before which we have something what we call it as bus stop talks. which means what we do is we introduce ourselves in less than 15 seconds each, and that way we can have a good amount of scientific camaraderie or collaborations. So that way anyone can really contact each other for any kind of things. So all of you please switch on your camera, and Hemani Sharma will take you to everyone based on the alphabetical order of our list. So and not more than 15 seconds, please. 15 seconds. So we are about 20. So it should not take more than about 4 minutes. Huh? Yeah. Thank you so much.
Thank you, sir. Good morning, everyone.
And one minute, Hemani.
Yes, sir.
One minute, I'm sorry. Yeah. Before which, can I request Harpreet to introduce Asif after this particular talk, please? Yeah. For Asif's talk. Huh? Thank you so much. Huh? Yeah. Yeah. Go ahead, Hemani. Yeah.
Thank you, sir. Good morning, everyone. Our keynote speakers have given a wonderful talk. Let's carry this wisdom forward as it's an opportunity for us to exchange ideas. I request each and every participant to give a small introduction. Let's begin with Barkha ma'am.
Yeah. Next, please. Yeah. Hemani, please. We don't have time. Yeah.
I request Miss Darshana Joshi. Please switch on your camera and give a small introduction.
Yeah, hello everyone. Myself, Dr. Darshana Joshi. I've done my PhD in Instrumentation and Control Engineering with specialization in Systems Biology for Type 2 Diabetes research. And I'm interested in expanding my work with the computational tools for various diseases and metabolic disorders. Thank you for arranging the session.
Thank you, Darshana Joshi. Now I request Bhanu Prakash Reddy. I request Gautam Manoj, please switch on your camera and give a small introduction for about 15 seconds.